Hello guys, welcome to my channel. So as you know, we went through the cell membrane if you saw the previous videos. So now we are going to be learning the new topic, that is protein structure and its function. And function. Right, so the protein structure and function, why the protein? So as you know, the protein is important for the cell to maintain the shape and the structure. But the protein are the main building blocks from which the cells are assembled, and they constitute the most of the cell dry mass. So in this topic, we will not only go through the protein that are related to the physiology, but we will go through the protein in the biochemistry as well. So we'll study a very deep, detailed topic for the protein structure and function. So on the hand, we'll cover biochemistry and the physiology as well. All right. Right. So as we know, proteins has many functions. For example, there are proteins like enzymes that promote intracellular chemical reactions. Then there are embedded proteins such as in cells that forms channels and pumps to transport or control the passage of nutrients and small molecules. There are some kind of protein that are signal integrators that relay information from plasma membrane to the nucleus of individual cells. There are specialized protein as well that are like such as an antibodies, toxins, hormones, antifreeze molecules, elastic fibers, or luminescence generators. So these are different kinds of protein functions. Okay, so on your screen you will see this pictures. So that is a few examples or the functions of protein that are really important. So that is one like the enzymes, as you can see in your picture. So it catalyzes the covalent bond breakage or formation. And there is an examples of as well, that is tryptophan synthetase or pepsin. So what the tryptophan synthetase do, it's make the amino acid tryptophan. All right, so that's an example of enzymes. So there is another function, that is structural protein. So that provide mechanical support to the cells and the tissues. So the examples you can see collagen and elastin. That is also one of the common type of the proteins. There are transport proteins as well that carry small molecules or ions. And the example you can see, that is serum albumin or hemoglobin that carries oxygen. All right, it's a good example. So the motor proteins that generate movements in the cells and the tissues. So that example is myosin. As you can see, there is a storage protein as well that store amino acids. So the example is small protein ferritin. There are signal proteins as well that carry extracellular signals from cell to cell. So the examples are insulin. You can see a small protein that controls glucose level in the blood. There is receptor protein as well. You can see that detect signals and transmit them to the cell response machinery. The example is rhodopsin. Right, there is gene regulatory proteins that bind to DNA to switch genes on or off. So the example is lactose repressor in bacteria silences the gene for the enzyme that degrade the sugar lactose. And there is special purpose protein that the functions are highly variables. In the examples you can see, that is for the plants and the fish. All right, so these are a few of the examples that we can see of the proteins. All right, right. So if you remember, or if you saw the previous video of protein where we discussed just the basics of it, so the proteins of every organism, from you can see from the simple bacteria or to the human beings, are constructed from the same set of 20 amino acids. So all the proteins that you can see, all different kinds of protein are constructed from same sets of 20 amino acids. These 20 amino acids are used to construct the protein. So like for example, the words or sentences that are now that is created from the 26 alphabet. So this is how the proteins are. So that you can see that's an alphabet for proteins, 20 amino acids. So from this 20 amino acids, the functions or the types of the protein we saw, that is enzymes, hormones, antibodies, transporter, muscle fibers, lens proteins of the eye, or whatever, that are constructed from this 20 amino acid. So this is amazing. Like each these 20 amino acids create different types of protein with different properties. And to generate a particular protein, amino acids are covalently linked in a characteristic linear sequence. So to understand the protein, we have to understand amino acids. So why? Because proteins are polymers of proteins are polymers of amino acids. All right. So, and like how this proteins are formed. So like each amino acid is covalently linked with each other to form the polymer, right? And the proteins, if there is a polymer of protein, it can be break down by hydrolysis, because when this amino acids join together, it loss of the element is the water. So like when they join, the water is expelled from it, right? So that is condensation reaction. So from all that 20 amino acids, so the first to be discovered was asparagine. So first amino acid discovered was asparagine, right? And that was in 1806, right? So, and the last of the 20, so the last one was 309. Last discovered. So this was discovered like around 1938. But the asparagine was the first one, and all the amino acids have common names, like in some cases derived from the source from which they were first isolated. For example, the asparagine that we are seeing here was first found in asparagus. So this was found in asparagus. So that's why it is called asparagine. And also another example is glutamate in wheat, that is gluten. So that's why it's called glutamate. So the other example is glutamate. Wheat or gluten. And tyrosine was first isolated from cheese. So that's why, because when we see the common name derived from the Greek cheese name, so that is called tyros. Tyros. Alright, so these are some of the common names. So all the 20 common amino acids are alpha amino acids. And why these are called alpha amino acid? Because it has an alpha carbon. So the common structure of amino acid is like the carbon that is in the middle, and this is an alpha carbon, and this carbon joins with an carboxylic group and an amino group, and there is a hydrogen bond, and this is R is the side chain. So this three structure will be the same except the side chain. So the amino acids or the proteins differ from each other in their side chain or these R groups, which vary in the structure, size, and electric charge, and which influences the solubility of the amino acids in water. And why this is called alpha carbon atom? Because the inorganic molecule, the first carbon atom that attaches to a functional group are referred to as an alpha carbon atom. That is why it is called alpha carbon, and that is why this is called an alpha amino acid. And this common amino acids of proteins have been assigned a three-letter abbreviation and one-letter symbol. For example, the glycine. So the abbreviation is GLY and the symbol is G. So this is how it goes on for all the amino acids. So for all the common amino acids except glycine, the alpha carbon, so this alpha carbon is bonded to the four different groups, right? The carboxylic group, the amino acid group, and the R group, and the hydrogen atom. But the glycine, so for glycine, the R group is another hydrogen atom. So for only glycine, but except glycine, all the other amino acids has this three groups and the side chain. So this alpha carbon atom is a chiral center. And what is a chiral center? So the chiral center is an atom that has four different groups that you can see bonded to it in such a manner that it has a non-superimposable mirror image. The mirror image of this is different. It is not the same. So why it is a chiral center? Because of the tetrahedral arrangement of this, of the bonding of the orbitals around the alpha carbon atom. So the four different groups that can occupy two unique spatial arrangement, and thus amino acids have two possible stereoisomers. So all these amino acids has two possible stereoisomers since they are non-superposable mirror images of each other. So these two forms represent a class of stereoisomers that is called enantiomers. So all molecules with a chiral center are also optically active, that is they rotate the plane polarized light. Right, so on your screen you can see the example of stereoisomerism in the alpha amino acid, that is for the alanine. So that is L-alanine and the D-alanine. All right, and these are just an example of the stereoisomerism. So the absolute configuration of the simple sugars and the amino acids are specified by the D and the L system based on the absolute configuration of the carbon sugar glycerol aldehyde, a convention proposed by Emil Fischer in 1891. So this means for all the chiral compounds, that is the stereoisomers having a configuration related to that of an L-glycerol aldehyde are designated as L, and stereoisomer related to D-glycerol aldehyde are designated as D. So this is the image that you can see on your picture for this. That's an example of the steric relationship of the stereoisomers of alanine to the absolute configuration of L and D glycerol aldehyde. So now the knowledge of chemical properties of the common amino acids is really important to understand the biochemistry. So what we can do, we can simplify this by just grouping the amino acids into five main classes, that is based on the properties of their R group, so this side chain, particularly like their polarity or the tendency to interact with the water at the biological pH. That is the 7. The polarity of R groups varies widely from non-polar and hydrophobic to highly polar and hydrophilic. But there are few amino acids as well that do not fit in this group, particularly glycine, histidine, and cysteine. Alright, so now we'll see the five classes that I mentioned. So the first class of the amino acid is non-polar and aliphatic R group. Alright, so in this group, but before going further, if you don't know what is aliphatic, so the aliphatic means, so if you know, there are like two organic compounds and they are aliphatic and aromatic. So there is aliphatic and aromatic compounds. So in aromatic compounds, so that means that have a benzene rings in this typical chemical structure that contains the 6 carbon atoms, while they're in an aliphatic compounds, they are either cyclics or linear structure, but like the aromatic is always cyclically bonded with alternating double bonds. All right, so that's the difference between the aliphatic and the aromatic, because there is one group that is aromatic as well. All right, so the non-polar aliphatic R groups. So in this class, amino acids are non-polar and hydrophobic. non-polar and hydrophobic. Right, so in this group, which amino acids are present? So they are uh, alanine, B, valine, leucine, and the iso leucine, E-methionine, F, there is proline as well. And there is one more. That is glycine. Alright, so 7. So this group contains 7 amino acids. So alanine, valine, leucine, isoleucine, methionine, proline, and glycine. Right, so the side chains of alanine, valine, leucine, and isoleucine tend to cluster together within the protein. So that stabilizes the structure through the hydrophobic effect because they have this property. Right, so but the glycine has the simplest structure. Right, although it is most easily grouped with non-polar amino acids, its very small size chain makes no real contribution interaction driven by the hydrophobic effect. And you can see the chemical formula or the chemical structure of this group as well. So the methionine is one of the two sulfur containing amino acid that has a slightly non-polar and thioether group in its side chain. The proline has an aliphatic side chain with a distinctive cyclic structure that you can see here. Right, so the next class is, so the first is non-polar aliphatic, the next is aromatic R group. Aromatic R group. Right, so this aromatic R groups has a phenylalanine, all right, then tyrosine, B and C, and then tryptophan. All right, so this aromatic R groups contain 3 amino acids, phenylalanine, tyrosine, and tryptophan, with their aromatic side chain, as you will see the image of this aromatic R groups in your screen, on your screen. So the hydroxyl group of tyrosine can form hydrogen bonds, and it is an important functional group in some of the enzymes. So you will see later. Right, and for aromatic R groups as well, these amino acids are relatively non-polar and hydrophobic. Right, so the third class is polar but uncharged R group. So these are polar and uncharged R group. Right, so this polar and uncharged R group, this group of amino acids are more soluble in water, so they are more hydrophilic. That means they are hydrophilic. All right, why? Because they contain a functional group that forms an hydrogen bonds with water. So this class of amino acids have what they have, let me list it here. So they have serine, right? So they have serine, then threonine, then cysteine, then I will do it here. Okay, I'll write it here, asparagine, and A is glutamine. Right. So this contains 5 amino acids: serine, threonine, cysteine, asparagine, and glutamine. All right. As these groups are polar group, so the polarity of serine and threonine is contributed by the hydroxyl group, as you can see the image, and that of the asparagine and glutamine are by their amide group. Right, and the cysteine, that is the one that why it is here, because its polarity contributed is by its sulfhydryl group, and that group is, so that's why that cysteine is really a weak acid, and that can make a weak hydrogen bonds with oxygen or nitrogen. So the asparagine and the glutamine are the amides of two amino acids also found in protein that are aspartate and glutamate respectively, to which this aspirin and glutamine are easily hydrolyzed by acid or base. So that as same as that cysteine is readily oxidized to form a covalently linked dimeric amino acid that is called cysteine, in which cysteine molecules or residues are joined by disulfide bond. So the disulfide linked residue are strongly hydrophobic, and the disulfide bonds play a special role in the structure of many proteins by forming covalent links between a part of polypeptide molecule or between the two different polypeptide chains. Right, so now the fourth class is positively charged. Okay, you can see that, right? Right, so, oops, sorry, positively charged R group. Alright, so that is just a basic R group, that is also called. So this has most hydrophilic R group are those that are either positive or negatively charged. Right, so the amino acids in which the R group have significant positive charge at pH 7 are lysine. Alright, so the one is, that is lysine. Then the second is arginine. Alright, and the third is histidine. Alright. Alright, so this group has three amino acids, lysine, arginine, and histidine, and you can see the chemical structure of this on your screen as well. And the last, fifth, is negatively charged or acidic group. Alright. Right, so this negatively charged acidic group has two amino acids, that is aspartate and glutamate. Alright, so this is the negatively charged acidic group. So this contains two amino acid that has a pH at pH 7 it is negative charge. Alright, and this has a second high carboxylic group on, as you can see on the chemical structure as well on your screen. Alright guys, I think this is enough for today. So this is a long topic though, so that's why we'll just go in the parts, but like for today this is enough. So we studied about the proteins and the 20 amino acids, and that are divided into 5 classes, and these are this 20 amino acids and the 5 classes listed here. Alright, thank you guys for watching, and don't forget to subscribe the channel as we'll be continuing with this study. Thank you guys.